All right, moving on to lesson 2 of chapter 20. It's called Prokaryotes. And our goal is to discuss distinguishing characteristics of domains and kingdoms of living organisms. Our objectives are to explain the two groups of prokaryotes differ, describe how prokaryotes vary in structure and function, and also explain the role of bacteria in the living world. Get rid of that for you. All right, let's think about it first. Imagine all your life as a member of what you believe is the only family on your street. Then, one morning you open the front door and discover houses and neighbors all around you. Where did all these people come from? What if the answer turned out to be that they had always been there? You just hadn't seen them. How would the view of your world change? When the microscope was first invented, we humans had such a shock. Far from being alone, we share every corner of our world with microorganisms. Even a seemingly clean toothbrush contains a film of bacteria on its bristles. Right there. So, objective 1 is explain how the two groups of prokaryotes differ. Let's talk about this now. All right, the smallest and most abundant microorganisms on Earth are prokaryotes, unicellular organisms that lack a nucleus. Prokaryotes have DNA, and like all other cells, but their DNA is not found in membrane-bound nuclear envelope as it is in eukaryotes. Prokaryote DNA is located in the cytoplasm. This is all review for you guys. A bacterium such as E. coli has a basic structure typical for most prokaryotes. Recently, biologists have divided prokaryotes into two very distinct groups, bacteria and archaea. These groups are very different from each other. Therefore, biologists now consider each group of prokaryotes as a separate domain. Eukaryotes are the third domain. The larger of the two domains of the prokaryotes is bacteria. Bacteria include a wide range of organisms with lifestyles so different that biologists do not exactly agree on how many phyla are needed to classify this group. Bacteria live almost everywhere in freshwater and saltwater and land and within the bodies of humans and other eukaryotes. E. coli, a typical bacterium that lives in the human intestines, is shown here. Bacteria are usually surrounded by a cell wall that protects the cell from injury and determines its shape. The cell walls of bacteria contain peptidoglycan, which is a polymer of sugars and amino acids that surround the cell membrane. Again, this is a review for you guys. Some bacteria, such as E. coli, have a second membrane outside the peptidoglycan wall that makes the cell especially resistant to damage too. In addition, some prokaryotes have flagella that they use for movement. Oh, what a nice clip there. There we go. Or pili, which in E. coli serve mainly to anchor the bacterium to a surface or to other bacteria. Under a microscope, archaea look very similar to bacteria. Both are equally small, lack nuclei, and have cell walls, but there are important differences. The walls of archaea lack peptidoglycan, and their membranes contain different lipids. The DNA sequences of archaea genes are more like those of eukaryotes than those of bacteria. Based on these observations, scientists have concluded that archaea and eukaryotes are related more closely to each other than they are to bacteria. Many archaea live in extremely harsh environments. One group of archaea produce methane gas and live in environments with little or no oxygen, such as thick mud and the digestive tracts of animals. Other archaea live in extremely salty environments, such as Utah's Great Salt Lakes, you might see right there, or in hot springs where temperatures approach the boiling point of water. So, explain how the two groups of prokaryotes differ. Prokaryotes are classified into bacteria or archaea, which are two of the three domains of life, and you need to know the differences and details between them. And objective 2 is to describe how prokaryotes vary in structure and function. All right, shape first. Prokaryotes range in size from 1 to 5 micrometers, making them much smaller than most eukaryotic cells. And prokaryotes come in a variety of shapes. Rod-shaped prokaryotes are called bacilli. Sphericals are called cocci. And the last kind, spiral and corkscrew-shaped prokaryotes are called spirillas. Prokaryotes can also be distinguished by whether they move and how they move. Some prokaryotes do not move at all, while others are prepared by flagellas. Some glide slowly along this layer of slime-like material that they actually secrete in front of them. Lovely thinkings. Prokaryotes need a supply of chemical energy, which they store in the form of molecules such as sugars. Energy is released from these fuels during cellular respiration, fermentation, or both. Prokaryotes vary in the ways that they obtain energy and the ways that they release it too. Looking at the two tables in the slide, which I do not see right now, notice that some species are able to change their method of energy capture or release depending on the conditions of their environment. Maybe I did not get that. There we go. That's what I should have had for you. So when a prokaryote has grown so that it has nearly double its size, it duplicates. Okay, there we go. It duplicates its DNA and then divides in half, producing two identical cells. This type of reproduction is known as binary fission. Because binary fission does not involve the exchange or recombination of genetic information, it is an asexual form of reproduction. When conditions are favorable, prokaryotes can grow and divide at an astonishing rates, some as often as once every 20 minutes. When growth conditions become unfavorable, many prokaryotic cells form what's called an endospore, a thick internal wall that encloses the DNA, almost like a nucleus, you might think, in the portion of the cytoplasm. Endospores can actually remain dormant for months or even years. The ability to form endospores makes it possible for some prokaryotes to survive very harsh conditions, and the bacterium Bacillus anthracis, which causes the disease anthrax, is one such bacterium. Mutations are one of the main ways prokaryotes evolved. Mutations are random changes in DNA that occur in all organisms, as you remember. In prokaryotes, mutations are inherited by daughter cells produced by binary fission. Many prokaryotes exchange genetic information by a process called conjugation, which we have talked about before a couple of times. During conjugation, a hollow bridge forms between two bacterial cells, and genetic material, usually in the form of plasmid, moves from one cell to another. Many plasmids carry genes that enable bacteria to survive in new environments or to resist antibiotics that might otherwise prove fatal. This transfer of genetic information increases genetic diversity in the population of prokaryotes. So objective 2 was to describe how prokaryotes vary in structure and function. Well, they vary in size and shape and the way they move and the way that they obtain and release energy. Objective 3 is explain the role of bacteria in the living world. Bacteria, called actinomycetes, are present in soil and rotting plant material such as fallen logs, where they decompose complex organic molecules into simpler molecules. By decomposing dead organisms, prokaryotes simply supply raw materials and thus help to maintain equilibrium in the environment. Bacterial decomposers are also essential to industrial sewage treatment, helping to produce purified water and chemicals that can be used as fertilizers. Cyanobacteria, right here, in the genus Anabaena, form filamentous chains in ponds and other aquatic environments where they perform photosynthesis. Photosynthetic prokaryotes are among the most important producers on the planet. Food chains everywhere are dependent upon prokaryotes as producers of food and biomass. As you remember from one of the very first lessons we had in the beginning of the school year, all organisms need nitrogen to make proteins and other molecules. Nitrogen gas makes up about 80% of the Earth's atmosphere, but only a few kinds of organisms, all of them prokaryotes, can actually convert the nitrogen gas into useful forms. The process of nitrogen fixation converts nitrogen gas into ammonia, and then ammonia can then be converted into nitrates that plants use or are attached to amino acids that all organisms use. Nitrogen-fixing bacteria and Archaea provide 90% of the nitrogen used by other organisms. Some plants have symbiotic relationship with nitrogen-fixing prokaryotes. The bacteria Rhizobium grows in nodules or knobs on the roots of, right here, you can see right there, legumes, such as clover, where they convert atmospheric nitrogen excuse me, into a form that is usable by plants. Bless me. Prokaryotes, especially bacteria, are used in the production of a wide variety of foods and other commercial products, like yogurt is produced by the bacterium Lactobacillus. Some bacteria can digest petroleum and remove, as you can see here, human-made waste and poisons from water. That's very handy for us. Other bacteria are used to synthesize drug and chemicals through the techniques of genetic engineering. Bacteria and Archaea adapt to extreme environments, may be a rich source of heat-stable enzymes that can be also used in medicines, food production, and industrial chemistry. So objective 3 is explain the role of bacteria in the living world. Prokaryotes are essential in maintaining every aspect of the ecological balance of the living world. In addition, some species have specific uses in human industry. To review our objectives, one was to explain how the two groups of prokaryotes differ, also describe how prokaryotes vary in structure and function, and then explain the role of bacteria in the living world. And they all support our goal to discuss distinguishing characteristics of the domains and kingdoms of living organisms. And there's a nice picture of a little plant. Have a good evening.